Eye is the organ of sight. It helps detect changes in the environment and dispatches signals to the central neural system. Human beings have a pair of spherical eyes located in eye sockets or orbits of the skull. The eyes are directed forward and adapted for binocular vision. These are protected by the eyebrows, eyelids, and lacrimal glands. The lacrimal glands secrete an aqueous solution that lubricates the eyes and helps rotate the eyeballs freely. The wall of the human eye is composed of 3 layers. The external layer, or sclera, is made of a dense connective tissue. The anterior portion of the sclera is the cornea. The middle layer, or choroid, is bluish in color and contains many blood vessels. The choroid layer is thin over the posterior 2/3 of the eyeball and thick in the anterior region, where it forms the ciliary body. The ciliary body continues to form the iris, the visible colored portion of the eye. The eyeball contains a transparent crystalline lens held in place by the ligaments of the ciliary body. In front of the lens, the iris surrounds an aperture called the pupil. The muscle fibers of the iris regulate the diameter of the pupil. The inner layer is the retina, which contains 3 layers of cells. The innermost layer is called ganglion cells. The middle layer is called bipolar cells, and the outermost layer is called photoreceptor cells. Photoreceptor cells are of 2 types, rods and cones. These cells contain photopigments. The cones are responsible for color vision and daylight vision, which is also known as photopic vision. The rods are responsible for twilight vision, which is also known as scotopic vision. A purple pigment called rhodopsin is present in the rods, which contains a derivative of vitamin A and is sensitive to dim light. However, the rods do not play any role in color vision. A violet pigment called iodopsin is present in the cones, which are sensitive to bright daylight or photopic vision. Cones are of 3 types and contain different photopigments and respond to red, green, and blue light radiations. Other colors are detected by simultaneous stimulation of more than one type of cones. Equal stimulation of all the 3 cones produces a sensation of white light. The cones are insensitive to dim light, so a person cannot see colors at night. The optical part of the retina contains 2 spots, known as the blind spot and the fovea. The blind spot is the region on the posterior pole of the eye, where the optic nerve leaves the eye and retinal blood vessel enters the organ. Photoreceptor cells are absent in this region, and therefore no image forms in the blind spot. The posterior end of the eyeball, lateral to the blind spot, has a yellowish pigmented spot called the macula lutea, with a central pit called the fovea. The density of cones in this region provides the best visual acuity. The aqueous chamber lies between the cornea and lens. It is filled with a thin, watery fluid, an aqueous humor that provides nutrition to the lens and cornea. The vitreous chamber is larger than the aqueous chamber and lies between the lens and retina. It is filled with a transparent gel called vitreous humor that gives shape to the eye, supports the retina and lens, refracts light rays, and maintains intraocular pressure inside the eye. The human eye works like a camera. The cornea, aqueous humor, lens, and vitreous humor all act as small lenses and refract light rays to focus on the retina. The iris acts as a diaphragm and regulates the amount of light entering the eye. Maximum refraction is done by the cornea and the lens brings final adjustment. Light radiations stimulate the photoreceptors on the retina and form a small and inverted image. But the object image is perceived in the right manner due to the interpretation of nerve impulses by the brain. The light rays in the visible wavelength focus on the retina to generate impulses in the rods and cones. The photopigments of the photoreceptors in the human eye are composed of opsin and retinal. Light dissociates retinal from opsin, which results in changing the structure of opsin. The structural change in opsin changes membrane permeability, which in turn results in potential differences in the photoreceptor cells. This results in the generation of action potential in the ganglion cells through the bipolar cells. The action potential or impulses are transmitted to the visual area of the cerebrum by the optic nerve. These nerve impulses are analyzed in the visual area and help to recognize the image formed.